Hello, everyone, and welcome to our live webinar. I am Heather Paich, and today I'll be sharing a presentation about compensation in flow cytometry. The objective of the presentation is to share some foundational knowledge about compensation upon which you can build in the future. This presentation should last around 30 minutes, and then I'll have some time afterward to answer a few questions if you have any. You can feel free to type your questions in the chat window. So here are the topics we're going to cover today. First, I'm going to share just a little bit of information about the ACEA Biosciences company. Then we'll jump into fluorescence and spillover, how to calculate compensation, single chain controls, and then compensation considerations and challenges. And then we'll have time at the end for questions. First, a bit about ACEA. ACEA Biosciences was founded back in 2004 on a technology called Excitation real-time cell analysis, and this uses impedance to monitor cells in real time. This technology has become very popular, especially with graduate students, because you can get tons and tons of data points with minimal upfront effort. Then in 2014, ACEA launched the NovoCyte flow cytometer, and then just this year, which is not actually shown on the graphic yet, ACEA launched our second flow cytometer called the Quanteon. So this is the NovoCyte, and it can analyze up to 15 fluorescent parameters, as well as forward scatter and side scatter, and it is customizable and field upgradable. The laser alignment and the timing are fixed, which allows for very consistent data from day to day, and there are no PMT pre-adjustments required for most applications, although you can do that if you want to. And then folks love the automated cleaning and shutdown features. And here's the Quanteon, our new flow cytometer. So it can analyze up to 25 fluorescent parameters, as well as forward scatter and side scatter. And this instrument utilizes silicone photomultipliers rather than the more traditional PMTs to detect light signal, and these have excellent sensitivity. It utilizes the same NovoExpress software as the NovoCyte, and that's considered to be both acquisition and analysis software. And the auto sampler can acquire from 24, 48, 96, and 384 well plates, as well as from standard fax tubes. All right, so let's move on to fluorescence and spillover. There are many different fluorochromes and dyes and stains out there that can be utilized in flow cytometry. And after being used to label cells or other particles, they can be excited by different laser lines, and then the emitted fluorescence light is collected. We often talk about excitation and emission maximums of particular fluorochromes, such as FITC. However, it's important to remember that these fluorochromes can be excited at wavelengths other than their maximum, and then more importantly for this presentation, fluorochromes emit fluorescent light over a range of wavelengths. Now this slide may look a little bit complicated, so I'll take a few moments to explain it. The figure shows the excitation and emission spectra for FITC. The excitation spectrum is shown with the blue dotted line, and FITC is maximally excited at 493 nanometers. We use the blue 488 laser line to excite FITC in flow cytometry. Then the FITC emission curve is shown in green, and the emission max is at 525. However, the full range of FITC goes from around 480 up to 650. In a traditional flow cytometer, we have to choose a filter set to select some of the emitted fluorescence of a given fluorochrome to be collected in a detector. And ideally, the maximum emission of the fluorochrome would be included in that filter set. It would be way too restrictive to use a filter set that would collect all of the emitted fluorescence from each fluorochrome. So in addition to giving some thought to what light our filter set choices will collect, we also need to give some thought to the entire range of the emission spectrum, particularly that light which is not collected in the intended detector. This figure shows the FITC emission spectrum in green, and then a detector that can be used to collect the FITC emission, which has a 530-30 filter. This means that the FITC detector will collect the FITC emission from 515 through 545 nanometers. However, we know that the FITC emission spectrum actually goes up to about 650. Therefore, some of the FITC emission will be collected in detectors intended to collect other fluorochromes. Here, it's being shown collected by the PE detector with a 585/40 filter and a PI detector with a 630/30 filter. The FITC fluorescence light collected in the PE and PI detectors is called spillover, also called fluorescence spillover or spectral overlap. And although we use strategies to minimize the effects that spillover might have in our experiments, it's a normal and expected part of flow cytometry. So let's take a closer look at what spillover looks like in some actual data. These data show a FITC single-stain control sample, which is, as you may have guessed, a sample stained with only FITC, and it's used as a control. This particular sample was created using compensation beads, which we'll talk more about later in the presentation. The bead population is gated on the scatter plot, and then all of the following plots will show only the events from the bead gate. The histogram below shows the FITC negative and the FITC positive events. Then these plots show the FITC spillover into the PE and PI channels. You can see the spillover in two different types of plots, the bivariate plots and then the histograms. We know that this is a FITC single-stain sample, so we expect there to be negative and positive FITC populations, but there shouldn't be any events that are positive for either PE or PI. So the populations in the bivariate plots that look like they could possibly be PE or PI positive are actually spillover from FITC into those channels. And we see something similar in the PE and PI histograms. The brighter peak in both of those peaks is the FITC spillover. Now, I'm going to ask you to remember these data because we will revisit them later in the presentation. So it's pretty straightforward to consider spillover of one fluorochrome into just a couple other detectors. However, we often use many fluorochromes in a flow cytometry panel. So here you can see some examples of commonly used fluorochromes, their spectra, and then potential lasers and filters that might be used. So at this point, I would like to mention that it's a best practice in the field to use single-stain controls to assess and control for spillover whenever you're using two or more fluorochromes in an experiment. And while it's really helpful to look at the theoretical emission spectra on the many awesome spectral viewers that are available, it's really important to test your fluorochromes on the instrument that you are using. The theoretical spectra do not always match the actual spectra, and because they can be influenced by many different factors. In addition, folks sometimes feel that they don't need to worry about spillover in experiments that only use fluorochromes excited by distinct lasers. So this is a great strategy, again, to minimize spillover. However, I would urge you to run the single-stain controls to double-check what's going on for yourself. Cross-laser excitation can occur, and it's important to understand how it might affect your data analysis. So to summarize, fluorochromes emit light over a range of wavelengths, and even though filters direct and limit the wavelengths of light collected in a certain detector, the light can still be collected by other detectors. And again, this is called fluorescence spillover or spectral overlap. Spillover can be corrected for mathematically, and this process is called compensation. So now we'll talk about calculating compensation. Calculating compensation has to do with the fluorochrome selected. However, it also has a lot to do with the specifics of the instrument you are using, including the laser lines available, the power of those lasers, and then the exact dichroic mirrors and bandpass filters that are being used to direct the light. So compensation is essential for multicolor experiments, and it prevents false positive and false negative data in your analysis. Compensation is calculated using single-stain controls, and these can be created using beads or cells. And as you can imagine, they only contain one single stain in each of the different controls. So a single stain should be created for every fluorochrome in the panel. And then just a few notes about compensation beads. There are many different suppliers of these beads, and they are used with fluorochrome conjugated antibodies. And the basic premise behind them is that there are beads that will bind everything, and there are beads that will bind nothing. So if you create a mixture of them and then add a single fluorochrome conjugated antibody that will bind to the bead type, then you've created a single stain control. And in addition, you can also have an unstained sample if you're going to be using that. So here's some requirements for single-chain controls. They should show clear, well-defined negative and positive populations if using the within sample negative population as a reference population. The other option is to use a universal negative sample, which can act as a reference negative population for all of the single-chain controls. For the purposes of this presentation, we're really going to focus on compensation that is calculated from within sample negative reference populations. However, you should be aware that there are other strategies out there to achieve the same objectives. In addition, autofluorescence should be the same for the positive and the negative population used to calculate the compensation for a specific fluorochrome. So if you are using the within sample negative reference population, you really don't need to worry about this so much. However, what it means is that you wouldn't want to use unstained beads as the reference sample for a positively stained cell population or vice versa. The positive population has to be as bright or brighter than your brightest experimental sample, and this is really important for compensation to be accurate. It's also important to use the exact same fluorochrome used in the experimental samples. It doesn't work just to pick a different fluorochrome that theoretically could be collected in that same detector and use it in place of the actual fluorochrome. And it's best to use the same tube of fluorochrome for your single-chain controls as your experimental samples. And this is really important for tandem fluorochromes like PE Cy7. Just one more note on this, it's a best practice to use the exact same fluorochrome conjugated antibody for your control samples as you use in your experimental samples. And for lowly expressed markers like some transcription factors or intracellular cytokines, some folks substitute a fluorochrome conjugated antibody with the same fluorochrome but directed towards a different highly expressed marker. And this can be problematic in some situations, particularly when using tandem dyes, and this is due to the potential differences in photobleaching. So, creating good compensation controls sometimes requires some thought, some creativity, and some work. And later on in the presentation, I'll share some problems and solutions for some common compensation challenges. So we're going to use FITC spillover into the PE detector as an example to discuss how to calculate compensation. Because flow cytometry is a relative science, without reference samples like unstained controls, it's really impossible to tell if a population is positive for any given marker. So I've added these dotted lines to this bivariate plot just for reference, like a thought experiment. Because this is a FITC single-chain control, we know there should only be FITC negative and FITC positive events. We know there should be no PE positive events, even though it might look like there are. So we need to adjust the FITC positive population downward so that it is more in line with the FITC negative population. And note that the FITC negative population is a reference population, so it's not adjusted. It's only the FITC positive population that is adjusted. So here's what it looks like after spillover has been accounted for. So again, it's important to note that we didn't just eyeball where the FITC positive and FITC negative populations are matching. We use actual measurements to make this determination, and we use the mean or median values of the two different populations. So now let's take a look at some actual data and some real numbers. This bivariate plot shows a FITC single-chain control before compensation, again, versus PE. So I drew the gates around the FITC negative and the FITC positive population so I could get some measurements. The median Y of the FITC negative population is around 1,000, while the FITC positive population is much higher, around 15,000. And I drew that orange dotted line at the FITC negative median Y for reference, because that's where we need to move the FITC positive population down to so that the medians match. We can do this in a number of different ways depending on which software you're using to analyze your data. Or you can always just let the auto compensation feature do the calculating and adjusting for you, which we usually recommend as a good first step. So this plot shows what the data look like after compensation. You can see that the median Y values are now matching, both around 1,000. So the FITC spillover into PE has been eliminated. And in this case, the value by which the spillover was reduced was 12.7%. So then let's consider the example data we looked at previously, where we noted the FITC spillover into the PE and PI channels, viewed in both the bivariate plots and the histograms. So this slide shows the before compensation data. And then this slide shows the after compensation data. You can see that the FITC negative and the FITC positive populations are evenly matched in the bivariate plots. And you can see that the brighter peak has completely disappeared from the PE and PI histograms. So the spillover, FITC spillover has been reduced by 12.7% from PE and then the 1.7% from PI. Now let's take a look at the before and after compensation data together. This slide shows the same plots we've been looking at, but in overlay format, my favorite. So the populations shown in blue are before compensation, and the populations shown in green are after compensation. So you can clearly see the adjustments that were made from the PE and PI channels. The FITC positive populations have shifted downward, and as a consequence, you can see that the data do appear to be more spread out. And it's important to note that there aren't really any changes in the FITC histogram. The adjustments we made did not alter the FITC signal into the FITC detector. So to summarize, how to calculate compensation, we use single chain controls to measure and then adjust for spillover into other channels by making the median or mean fluorescence of the positive equal to the negative. The value we get in this adjustment is a percentage of fluorescence to be subtracted for that correction. Then the percentage is calculated from looking at all the different single chain controls into all the other different channels are compiled into one compensation matrix, and then this is applied to the experimental samples for analysis. Here are two different compensation matrix examples, one with only 3 fluorochromes and one with 6. However, I'm sure you can imagine just how complex things might get when using 12 or 15 or even more fluorochromes. So now we're going to move on to discuss some more specifics about single chain controls, or as I like to say, cells versus beads. So in flow cytometry circles, there seems to be some really strong feelings about using either cells or beads to create the optimal single chain controls. Two different camps, you might say. And although I'm firmly set in one of those camps, I'm going to do my best to share objectively some advantages, disadvantages, and considerations of each so that you can determine which is the best fit for you and your projects. First, we will consider using beads to create single chain controls. Beads work well no matter the biological expression level of your markers of interest. For example, markers that are rare, such as FOXP3 on T regulatory cells, can be challenging to use as single chain controls because of the potentially low levels of positive events. Using beads can circumvent this challenge. In addition, beads usually provide more clearly defined and better separated negative and positive populations. And this is especially true when your marker of interest may be lowly or variably expressed, or if your marker of interest does not show a bimodal distribution, meaning as in a clear yes or clear no pattern, and is rather expressed on a continuum. So beads work well for high color panels, especially because bead samples might be more straightforward to prepare than cell samples, and in addition, they're going to conserve your cells for your experimental samples. However, beads are not biological, so if that is important in your particular model or your experiment, you may need to consider how that would affect your outcomes of interest and then potentially shift towards using cells. Finally, bead samples can be often used for perhaps about a week if they're stored properly, although this isn't ideal if you're using tandem fluorochromes because of the stability concerns. Next, we'll consider using cells to create single chain controls. So cells work well if all the markers are highly expressed, especially if they show a bimodal distribution with that clear positive or negative signal. Cells are great if you are using a smaller number of fluorochromes in your panel, or if your fluorochromes are generally spread out across distinct lasers. Preparing cell samples can be more work if the cells need to be stimulated to express a particular marker, especially if that marker is only expressed at certain time periods, for example, early or late activation markers, or If the marker is an intracellular cytokine or a phosphorylated protein, additional steps will need to be accounted for in the sample preparation, of course. And cells can help account for autofluorescence, and this is more of a concern with some cell types than others, but certainly something important to consider. So hopefully that gives you some aspects to think about when determining how to best create your single-chain controls. And it's worth mentioning that folks often may need to use a combination of cells and beads to create the ideal package of single-chain controls for their experiment. So now we've talked about fluorescence spillover, how to create single-chain controls, and how to calculate compensation. Now I'd like to share a couple of compensation calculation considerations. First, let's assume you've run all your single-chain controls and you've allowed the auto compensation feature in your analysis software to calculate and apply compensation. What happens next? So I would suggest that what comes next is a visual double check of all your single-chain control samples. So it's a good idea to look at the scatter plot to make sure that the gate has been drawn on the optimal population. And this is especially important if you're using auto gating, because sometimes you may want to use your human logic to override the computer logic. You also want to take a look at the histogram where the positive and negative populations are identified. And this is really important because these are the populations that are used to do the actual compensation calculations with the medians or means. Then you want to look through all the bivariate plots to make sure that there are no instances of over or under compensation, and also that there's no contamination present in the sample. And if you do see areas of concern, it's really better to address them before compensation is applied to the experimental samples. So in looking at the data on this slide, it's pretty straightforward and it looks pretty good visually. And later on, I'll show some examples of some serious compensation challenges. So I've just mentioned how valuable visually double checking through your compensation data can be, but there are times when visualization alone can mislead you, and I'll show you a couple examples next. So this slide shows the same FITC single-chain control sample that we've been working with, and all the plots are FITC versus PE. The first panel has no compensation, and then the second panel has the optimal compensation applied. However, the third and fourth panels both show undercompensation. The third panel is very clearly undercompensated. We can see that just by a quick visual check. However, the undercompensation in the fourth panel is not quite as obvious. Without looking at the actual numbers, the median Y values of the FITC negative and positive populations, we might not notice the undercompensation as readily. And I think that this slide shares a really important message because I have often heard folks say that they just eyeball or estimate the compensation based on what things look like visually. However, it's actually much more accurate to trust the auto compensation to do the calculations for you, or else if you actually draw the gates around the negative and positive populations to observe the median or mean values while you're making your spillover adjustments, that's much better. This slide shows some similar data, but for overcompensation. So again, the first panel is no compensation, the second panel is the optimal, and then the data in the third panel is very easily identified as being overcompensated. It looks horrible, while the data in the fourth panel doesn't look too bad visually. However, when we actually look at the median values, you can see that it's not optimal. So improper compensation can introduce unwanted error into your data analysis. So I would urge you not to eyeball it or estimate it, and I would urge you to take the time to make sure your compensation matrix is accurate before you apply it to your experimental samples. Finally, we're going to look at some common compensation challenges. This sample is intended as a FITC CD3 single-chain control. Because of the amount of debris that is present in the sample relative to the number of cellular events, the auto-gating feature has drawn that main gate on the debris rather on the populations of interest. And this is because it often gates on the most dense area of events, so you've got to be careful of this. And the solution is to redraw the main gate to include the cellular populations. And In this particular sample, it's also going to be important to include both the lymphocytes as well as the monocytes, macrophages, et cetera, in the main gate, so that there's going to be both CD3 negative and CD3 positive populations represented in the histogram. This sample is intended as a live apoptotic cell discriminator using a Nexin V FITC, which stains apoptotic cells. Unfortunately, this is a very healthy cell sample, so there are no Annexin V FITC positive events. As such, this sample cannot be used to calculate compensation accurately. The solution has to be to create a new single stain control that contains both apoptotic and live cells. So there would be a well-defined positive and a well-defined negative. Now, this is a situation where you might be super happy that you have lots of healthy cells with very few dying cells, and this may be ideal for your experiment, but it is not ideal for your single stain controls. So it does take some work to create good single stain controls, and in this case, you might need to divide your cell sample into two parts. So kill one part and keep the other part happy, and then combine the two before adding the stain. This sample is also intended as a live apoptotic cell discriminator using a Nexin V FITC, and based on the scatter plot, we can guess that there are both live and apoptotic cells present in the sample. However, when we look at the first two histograms, we don't really see a well-defined positive population in the first or a well-defined negative population in the second. And the problem is with where the main gate is drawn. The first panel excludes the presumed apoptotic cells, and in the second panel, it excludes the presumed live cells. So the solution is to draw a gate that includes both populations to get two well-defined populations on the histogram, and you can see that in the third panel. So it's important to note that the gating strategy that is used in the single stain controls does not have to match the gating strategy you use in your experimental samples. Depending on your scientific question, you may actually want to draw the main gate on the presumed live population on the scatter plot and then drill down into the other populations of interest in your actual samples. However, the objective of the single stain controls is to calculate an accurate compensation matrix. So you may need to use some creativity to achieve that, and I think that's one of the things that makes flow cytometry so much fun. This sample is intended as a FITC single stain control marker unknown, and this is a bead sample, and it looks like it contains some doublets and clumps. The problem, which is not a huge issue, but could be addressed if you were interested, is that the doublets and clumps are included in the main gate, and this makes the histogram not quite as sharp as it could be. So the solution, which you can see on the bottom, is to change the main gate to include only the singlet beads. And you can see that cleans up the histogram significantly. This sample is intended as a per-CP single stain control, again, marker unknown. And the problem is that there's not much separation between the negative and the positive populations on the histogram. And another potential challenge is that the positive population may not be as bright or brighter than all of the other experimental samples. So one possible solution is to create a new per-CP single stain control sample using beads and reacquire the data. This would allow for better separation between the two populations and would likely provide a higher brightness of the positive population. This sample is intended as a FITC CD19 single stain control. We can tell by looking at the bivariate plot that it is not a true single stain control. Another fluorochrome, live dead aqua, was inadvertently added. So the most straightforward solution to this challenge would be to create and acquire a new FITC CD19 single stain control that contains only the FITC. This sample is intended as a PE-Cy5 CD14 single stain control. The first two times the main gate is drawn on the scatter plot, there really aren't that many CD14 positive events in the histograms. So the solution is to redraw the main gate to enrich for the CD14 positive population to increase the number of positive events, which is going to increase the robustness of the compensation calculation. And again, this may be not exactly how we want to gate for our experimental analysis, but that's okay. We may need to be a little bit creative to create the optimal compensation matrix. So to summarize, it's important to create good single-chain control samples in order to calculate compensation. As scientists, we focus so much on our experimental control samples, understandably, and we want to do that to make sure we're able to effectively answer the questions we're asking. But when flow cytometry is the tool used to answer those questions, there's a whole additional set of controls that need to be considered. So you may need to get a little creative in preparing your samples and in gating your data. Remember, the gating strategy used for the single-chain controls can be completely different from the one you use for your experimental samples. Finally, I would like to end by sharing that I recognize in real life we sometimes have to make choices to do our science, and we're not always able to follow best practices to a T. So I hope the information that I've shared with you today will allow you to at least make informed choices about single-chain controls in your future flow cytometry experiments. So I could probably talk for a couple more hours about compensation, and we've really just scratched the surface of everything compensation related, but I hope it gives you a solid foundation on which to build. There are a lot of excellent resources out there, and of course, ACEA will be presenting additional webinars in the future that you might find useful. So let's open the floor up to any questions you might have. Okay, I've got a question that says, can I have this PowerPoint? And just to let you know, we are going to be publishing this webinar on the ACEA Biosciences website, probably in a week or two, so you'll be able to access the recording there. All right, and I have another question. I've heard that values over 100% in the compensation matrix are bad. What do you think? Okay, so this is kind of another controversial area in flow cytometry. It's a pretty deeply rooted edict of flow cytometry that compensation values over 100% are not good. And I completely understand where that comes from. However, I would say that as we've added more fluorochromes and more lasers to our repertoire, and as the engineering with the instruments themselves has evolved, as well as the software and hardware that process the data, and even the improvements in dynamic range and scaling, we should really allow for a little more leeway in handling large percentages in the compensation matrix. And I mentioned briefly during the presentation about data spreading. And data spreading, higher degrees of data spreading are certainly associated with higher compensation values, and it is an important consideration. However, I would argue that the objective of running flow cytometry analysis is not necessarily to draw perfect pictures, even though that's fun. I would say that the objective is to generate high quality data that can confidently answer your specific scientific questions. So I would say that concerns about data spreading should be application and panel dependent. You want to be confident in your ability to accurately identify your populations of interest. So if data spreading interferes with it, it is definitely a concern. But if you can get good data from an imperfect plot, I think you're still achieving your objective. Thanks for that question. So I have another question that says, I used to hear about the isotype staining. Is it mandatory to have good compensation? So this is a good question, and there are so many different types of controls in flow cytometry. So isotype staining is really not associated with compensation or single chain controls used to calculate compensation. Isotype staining is really used to check for nonspecific binding, and then historically it's been used as a gating control, although the field has been moving towards FMO controls or fluorescence -1 controls for more appropriate gating controls. So yeah, isotype staining is not really associated with the compensation. All right, and I have another question. I am using a cell line in which all the cells express GFP. How can I create a single chain control for GFP to use in compensation? So this is a good question. I actually hear it a lot for GFP as well as the other fluorescent proteins. So at least for GFP, I know that Thermo Fisher and possibly some other companies offer GFP beads, and those can be used for compensation, and that's a great option. For other fluorescent proteins, it can be a little bit more challenging if all of your cells express that fluorescent protein. So I think the best advice that I can offer is to use cells from a similar cell line that does not express GFP. And by similar, I really mean similar autofluorescence, which, you know, it's kind of hard to assess your autofluorescence in your cells that do express GFP, but one way to get around that is to look at where your GFP expressing cells fall in the channels that should have minimal spillover from GFP, and then you can use that to match it up with the cells that you would use for your negative control. Good question. All right. I don't see any other questions, so I think we are going to wrap things up. Thank you all so much for attending and for your participation, and best of luck in your future flow cytometry experiments. Thanks so much. Goodbye.